All right, so last week we learned about what's in our genome, mostly the human genome, and we talked about the types of genes that we have and the types of repeat DNA that we have, and emphasized the fact that a large majority of our genome is composed of the repeats, and how these repeats actually jump from one side to another, and what kind of repeats that we have, focusing a lot on the transposons. And today we are going to spend some time talking about how the genome is packaged inside the nucleus. So it's a cliche now to say that the human genome is about 2 meters long if you stretch the DNA out from one end to the other, and that actually has to fit into a nucleus of 10 micron in size. And it has to be done not haphazardly, because when it's fit inside the nucleus, it still needs to give access to proteins that are going to use the information in the genome, such as the transcription machinery, replication machinery, repair machinery, right? So this packaging needs to be dynamic, and it needs to be accessible. And we know that the packaging and the way that the DNA is packaged in the nucleus actually regulates these processes, okay? So today we're going to talk about these basic molecular mechanisms by which the DNA inside the nucleus is packaged in eukaryotes. All right. So I'm going to start this very classic picture that was taken by Walter Fleming, who was a German biologist more than 100 years ago, who was looking at the cell division in salamander fins and gills. And what he noticed was that every time a cell divides, he sees these thread-like structures. And he coined the term mitosis for the cell division, and he described these chromosomes. At that time, he didn't know that these chromosomes actually carried the genes and the hereditary information. He was basically making a structural observation. Even to date, you will hear descriptions of chromosomes as thread-like structures. We want to actually move away from those visual descriptions to molecular descriptions. So read your textbook and see what are the modern descriptions of chromosomes nowadays, okay? And these thread-like structures are the linear eukaryotic chromosomes. They carry, they're basically the DNA. And what they're made up of is a mixture of DNA, protein, and RNA. And this mixture we call the chromatin. Okay? All right. So that picture should give you immediately idea that the chromosome structure is dynamic during cell division. What you see is an interface. So this is a picture of an interface nucleus. What you can see here, it's the nucleus right there, stained by a dye that stains the more condensed regions of the nucleus a little bit darker than others. What you can see here is that you see a round nucleus, and you see these dark spots which turn out to be heterochromatin. These heterochromatin are very condensed chromatin structures where there is not much activity, such as transcription. And then you see these lighter spots which are corresponding to euchromatin, where the DNA is more active. And one thing that you might notice is that you actually cannot see individual thread-like chromosomes, right? And the reason for that is the chromosomes are decondensed during interphase. Interphase is the time of the cell cycle where it's not mitosis, okay? During mitosis, the nuclear envelope breaks down, typically in many of the eukaryotes, and what you can see is these typical X-shaped structures that would come to your mind when you think of chromosome right away. But this type of structure is only in mitosis, where you can see these condensed chromosomes, okay? And at this stage, the chromosomes are tightly packed and individualized, so you can actually visualize them. All right, how does a mitotic chromosome look like? Let's just give you the features of the metaphase chromosome. So metaphase is the stage of mitosis where the chromosomes are in their most condensed state. So metaphase chromosomes look like this in a eukaryote, and it has an X shape. So the ends of the chromosomes are called the telomeres. The region of the chromosome which attach to mitotic spindles is called the centromere, and is required for segregation. So at this stage of the cell cycle, why are we seeing this structure? Are we looking at one chromosome or two chromosomes? Raise your right hand if you think it's one. Raise your left hand if you think it's two. There are two chromosomes here. They are sister chromatids. So the reason for why we are looking at two chromosomes is mitosis is at the stage where you have where each chromosome is replicated. Okay, so what happens during the S phase is you have a single linear chromosome, and during the S phase, this then gets replicated. Now you have two copies of that chromosome. So we're looking at two chromosomes. These are two sister chromatids. And during the S phase, these two sister chromatids are held together by a protein complex called cohesin, which forms these ring structures and keeps these sister chromatids together. Okay? When these chromosomes, when the cell is ready to go through mitosis, these cohesin rings are removed from the chromosome arms. Okay, so what you're going to have is you're going to have these cohesin rings removed, except at the centromere. This centromere is held together by the cohesin complex, because what you want to do is you want to attach the centromere to spindles from opposite poles, right? So that during mitosis, these chromosomes can segregate into the two daughter cells. And this is the mechanistic reason why the mitotic chromosomes show this X type of shape. Is that clear to everyone? Okay. All right. So because the mitotic chromosomes are easy to visualize, it's very hard to visualize the interface chromosomes. The mitotic chromosome structure has been used to look at the karyotype of a person or of an organism. That is, the karyotype is the number, size, and shape of chromosomes in an individual. Okay? And the karyotype is unique for each species. So way back when people were deciding if something was two different species, they would look at the numbers of chromosomes, shape and size of the chromosomes during mitosis to distinguish between species. So the traditional methods to look at mitotic chromosomes involve GEMSA staining. This is basically a permanent dye that binds to DNA after the chromosomes are subjected to mild heat or proteolysis. Okay, and this gives yields to the G bands, which I'm going to show you a picture of. There are now more modern methods that use fluorescence in situ hybridization, and this is based on hybridization of fluorescently labeled probes specific to different regions of the chromosomes. Okay? So first, how does the GEMSA staining look like? This is the human genome, mitotic chromosomes that are stained by GEMSA. And what you can see is that somebody who is an expert actually went through each of these pictures and decided by looking at the size and the shape and the banding pattern on each chromosome and labeled them 6, 19, 18, etc. I wouldn't be able to do this, right? But these are trained people that know that the GEMSA pattern is going to indicate what chromosome it is. What's important is that patterning is reproducible. That means every time that these people are going to stain the mitotic chromosome, they're going to see the exact staining pattern for a given chromosome. And it's going to tell you the identity of that chromosome. How do we do this these days? What we usually use is fluorescence in situ hybridization. Okay? So the way that this works, let me see if I can draw it. So what you start with is you have your chromosome. Let's just simply draw it as a double-stranded DNA. Okay? And you want to visualize this chromosome. What you first do is you generate a probe. A probe is a piece of DNA that you're going to label. It could be radioactive. It could be fluorescence. In this case, it's going to be fluorescence. Oh, okay? So you're going to generate this probe in many different ways. One of the ways which you can generate this is by PCR, which you've learned, right? So what you can do is you can design two primers. Let's say we want to visualize this side of the chromosome that is going to amplify a certain sequence. Okay? So in your PCR reaction, what you're going to do is you're going to put your genomic template, you're going to put your PCR primers, right? And you're going to add DNTPs. Now, these DNTPs, you can buy special ones that are chemically conjugated to certain fluorophores. Fluorophore means some chemical that's going to give some kind of color, right? So what you can do is you can basically use DNTPs that are conjugated, for example, to red. Okay. Once you do your PCR, what is going to happen is you're going to generate a probe, which means lots of copies of the same DNA fragment that is going to contain these red DNTPs incorporated, right? And these are going to appear red under a microscope. Does that make sense so far? Okay, so what you do next is you take these probes and you take them and you first denature the DNA so the two strands are separated, okay? And then next you hybridize. Hybridize means you incubate your probe or whatever that you're going to trying to hybridize to the genome. You're going to mix your chromosomes with the labeled probes in a denatured solution, and then you're going to change the conditions so that DNA can complementarily anneal to each other. Okay? So now this probe is actually complementary to this region of the chromosome, correct? So what is going to happen is that the probes are going to align, hybridize to their corresponding region. Okay? And because these are red, when you look under the microscope, that region of the chromosome is going to appear red. Does that make sense? This is an important concept that is going to be used over and over again in molecular techniques. So make sure you understand this. Any question? Okay. All right. So basically what you have here is that you have a fluorescently labeled probe that you're going to denature and hybridize to your target DNA. And that target DNA site is going to now look whatever your probe color is under the microscope, okay? So when you do this with a combination of probes that you generate for the whole genome, so you generate hundreds of different probes, and you use hundreds of different of fluorescently conjugated label combinations, what you can do is you can basically analyze the whole genome, and that's what this is. This is called spectral karyotyping, okay? So instead of looking at just one region with a red probe, what you do is you make lots of different probes and look at the whole genome. So in these cases, the probes are made by DNTPs that have different combinations of fluorophores, okay? And then the microscope is going to take these images. Then an image processing software is going to decide what probe corresponds to what color, okay, and say, okay, this region is chromosome 1, left arm. This region is chromosome 2, right arm, etc., etc. And then what it is going to do is it's going to give generate an image of each chromosome, and at the end, what you're going to see is a karyotype of a mitotic of the cell that you're analyzing. Karyotype tells you a lot, right? In humans, we said that we have 46 chromosomes. So when you see this many of chromosomes, and based on the shapes and the sizes of chromosomes, you can say that this is coming from a human, right? And there's another thing that you can tell right away in humans from the karyotype of a person. What could that be?
The X chromosome?
Yeah. This person is a female because it has 2 X chromosomes rather than an X and a Y, which would be a male, correct? All right. So what are the different levels of DNA compaction that you can achieve with different mechanisms? Here is just a summary of this, going from the naked DNA. So this is the DNA double helix. it's about 2 nanometers in width. The first level of compaction is wrapping this DNA around the histone proteins to form nucleosomes. Okay, and this structure is called beads on a string structure. It has a width of about 11 nanometers. So from here to here, you already compacted the DNA somewhat, right? And then these beads on a string form of chromatin basically folds within itself and forms more and more compact structures that are thicker and thicker, right? And these chromatin fibers, we call them, fold into further structures, finally giving rise to the mitotic chromosome, which is 10,000-fold shorter than the initial double-stranded DNA. Okay? So how do these actually look under microscope? These kind of models are produced based on pictures that people look at chromatin under the microscope and molecular experiments where you interpret the results and come up with these kind of models. Okay? At the very basic level, you can actually look at a DNA double helix using atomic force microscopy, which basically is not very exciting, right? So it's basically kind of like a thread. So this is where things get really exciting. If you isolate chromatin from nuclei and look at under the microscope under conditions where these beads on a string can be revealed, you can actually see them. So here's a bead, and each of these beads is a nucleosome, and you can see the string, which is the DNA that's in between them, right? And these beads on a string chromatin condenses further. You can actually see that more thicker fragments are actually made. This resolution here, we don't have very many pictures. It looks like the chromatin is very dynamic. It can change from loops to strings versus more condensed chromatin, so there aren't many pictures of it. But there are pictures of mitotic chromosomes where you can see that the whole chromosome is basically condensed in these global structures. Like it's a blob here, it's a blob here, etc. And this is one chromatid, and this is the other chromatid. And if you actually take this mitotic chromosome and kind of squish that on a slide, what you can see is that this structure, which is a proteinaceous structure that holds the DNA together in mitotic chromosome, which is also referred to as mitotic scaffold. All right. So the interface, the level of compaction that we see typically are here. So what you see in interface is you're going to have naked DNA. Why do we have naked DNA in the interface? If you took a snapshot of an interface nuclei, you will always find some naked DNA. What could be the reason for that? What's happening in interface? What is the genome used for? What molecular machineries use the genome? Come on, guys. It's Monday, I know. Transcription. Can transcription happen on condensed DNA? No. What it has to do, it has to unwrap the two DNA strands and use one of them as a template to make the RNA, right? So in a region of the genome where there is active transcription, you expect to find some form of naked DNA, right? All right. So in interface, we have these lower levels of compaction, and that's dynamic, and that means that not every part of the genome is going to show the same level of compaction, and not all at the same time. That is, if a gene is transcribed, it's going to be more open. If it's not transcribed in another cell type, it's going to be closed, right? So what you're going to see in interface is you're going to have a mixture of these forms up to these chromatin loops. And in interface, and what we refer to these type of structures is that is the complex of the genomic DNA with RNA and protein in the nucleus is chromatin. All right. So the basic repeat structure in chromatin is called a nucleosome. Okay? Nucleosomes are basically these beads in the bead on a string structure. They are made up of DNA plus histone proteins. The histone proteins are named here histone H2A, H2B, H3, and H4. These are small basic proteins that come together in an octamer made up of 2 copies of each histone. Okay? So that histone octamer, here's a crystal structure of that octamer. So 8 histone proteins here, 2 copies of each, and they come here together, form this very stable hockey puck-like structure. Okay? So then the DNA wraps around that structure about 1.8 fold turn. So it wraps around this structure, and the amount of DNA, the length of the DNA that wraps around this is about 147 base pair. So in terms of terminology, the protein here that is called the histone core, okay, this is called the histone core, histone octamer, because there are 8 copies, and the DNA around it is called the nucleosomal DNA. The DNA plus the histones, this whole structure is called a nucleosome. Okay? And the nucleosomes are the basic repeating units of chromatin. In our cells, typically the nucleosomes the nucleosomes are bound by another histone called histone H1. This histone is called the linker histone. It's very abundant, just like the regular histones. But it's not in the core of the nucleosome. Where it is it binds to the linker DNA. Okay, the linker DNA is the DNA that is in between these beads on a string. So this DNA that links the 2 nucleosomes is called the linker DNA. Okay, and the linker histone H1 is very abundant and binds to that linker DNA. And what histone H1 is predicted to do is that it basically compacts the nucleosomes a little bit more. So you find histone H1 at regions of the genome where there is not much activity, such as transcription. Okay, so when the nucleosome was crystallized in 1998, and people analyzed the structure, what they found is that the tails of the nucleosomes, the N-terminal tail especially, that is the beginning of the histones, the N-terminal tails of the histones were not in the crystal structure because they could not see it in the crystal structure because they were flopping around, they were dynamic, okay? They weren't all set in place where you can see in an X-ray crystallography. So these N-terminal tails of the histones are sticking out from the nucleosome. Okay, it's flopping around and doing all kinds of things. And a few years later, research in the next 10 years or so established that the histone tails are post-translationally modified. What that means is that histones are made in the cytoplasm, right, just like any old protein, and then it's shuttled back into the nucleus. It's put onto the genome, so the histones are deposited into the DNA, the nuclear DNA, and then there are protein complexes that come in and modify the amino acids on the histone tails to add certain modifications. Okay? And these modifications are called the post-translational modifications on histones. They typically happen on the tails that are accessible. So here in the crystal structure, what people have done is just to give you a sense of where these tails might be. They put some dashed lines for how the tails are sticking out, right? And if you think of the nucleosome like a hockey puck, right? I always think of it as such, right? So when you look at from the face, you see this nice and round structure. You see the octamer and the DNA wrapping around. But when you flip it to the side, it looks like a little hockey puck, right? And the tails are sticking around from that. So the types of modifications, post-translational modifications that occur on the tails are various. One of the earliest one that was discovered is called acetylation. So you basically put an acetyl group. Later we found that histone tails can be methylated and phosphorylated and ubiquitinated. And as the mass spectroscopy experiments are getting better and better and having more and more sensitivity, we found that the histones can be actually modified by other chemical groups such as sumolation, biotinylation, crotonylation, hydroxylation, etc. Okay? And what we found is that the tails are where the modifications mostly occur, but there are also some modifications that can occur in the core region of the histones. Any questions about this? Does everyone understand what post-translational modification is? Okay. So these modifications are not just for fun. Okay, they have some kind of function. And the modifications are put, they are read, and they're erased. Okay, so think of these kind of information on top of the DNA sequence. Okay, so how you can conceptualize these histone modifications and the mechanisms that regulate these modifications is that you can think of these as written, read, and erased. Okay, so the writing occurs after the histones are deposited in the genome. So you have a nucleosome right here. You have the histone tails are sticking out. And I said that there are protein complexes containing enzymes that are going to modify. That means just add a little chemical a molecule to these tails. And these enzymes can come in different flavors. And one of this is called an acetylase. So it's going to acetylate a certain amino acid on the tail. Other one could be a methylase. Another one could be a phosphorylase. It's going to put a phosphor group onto the tail, okay, and onto a certain specific amino acid on the tail, and that's important. So each of these have a specificity. The enzymes that erase is very, it's basically the enzymes that remove these groups off of those amino acids. So an eraser of an acetyl group is going to be called a deacetylase. An eraser of a methyl group is going to be called a demethylase. And an eraser of a phosphate group is going to be called a phosphatase, right? All right. So this is writing and erasing. Now, when these modifications are on the tails, they are bound by proteins that have certain protein domains. These are certain regions of the protein with a structure that's going to be able to dock onto these modifications. Okay? And these are called the reading proteins. So these proteins are going to read the modification. Let's imagine that this little blue ball is a certain type of modification. It could be methylation, right? So it's going to be put up here by a methylase. It can be removed by a demethylase. But let's imagine that when that methylation is put on the histone tail and there is this protein called a reader, we'll have a domain that's going to recognize this methylation. And these type of methyl reading domains could be a PhD finger or a chromodomain. And this protein is going to bind to these nucleosomes, right? And these proteins are called the readers, and that's how the information is read. The information is read through protein domains. Bromodomains read acetyl groups. Chromodomains and PhD fingers read methyl groups, and as well as WD40 repeat. So certain chromatin proteins with these groups are going to then go to regions of the chromosomes where these modifications occur. This is important is an important concept. What it is telling you is that it's giving you a mechanism by which you can target a protein to a region of the genome, right, that's actually not using DNA sequence. Can you tell me a mechanism by which a protein can be targeted to a certain region of the genome through using DNA sequence? transcription factors, DNA binding transcription factors. These transcription factors recognize short DNA sequences and go to the regions of the genome that contain these sequences, right? And here is a mechanism where you're not using the DNA sequence. You're actually using the histone modifications on the nucleosomes that are wrapping the DNA around. Any questions about this?
So when it's written erase or written read and erased, is this happening multiple times?
Yes, it is a very dynamic process. And the dynamics of each process depends on the type of the enzyme and the type of modification. For example, acetylation is very dynamic. So acetylation usually opens up chromatin for transcription. We're going to talk about that in more detail in a few lectures. But it happens, and the acetylases remove it constantly. Methylation is a different story. Metal transferases put the methylation, but these methyl groups can stay for a very long time because the demethylases aren't as active. They're usually activated at certain regions of the genome at certain times during development. So how long these modifications are going to stay is going to be a result of how active is the writer, how active is the eraser, right? Any other questions? All right. What do they actually do, right? What is the function of these histone modifications? There are different types of functions that I'm going to basically just classify into three main categories. In one category, a modification on a histone tail can actually change the way that the chromatin is compacted, okay? So, for example, acetylation of histone tails cause them to be a little less compacted. These histone modifications can inhibit binding of certain proteins. Let's say the methylation of the histone tails can prevent a certain protein that actually likes tails without that methylation. So in that case, these kind of modifications can kick out a protein that's bound to chromatin, right? Or it can actually facilitate binding of certain proteins. In this case, the modification helps a certain protein to bind to chromatin, right? So now we just learned reader, writer, or eraser. What is the category that you would put this modification binder protein, mod binder? What category would you put that? Reader, right? Because it reads, it binds to it. A writer is actually the protein that's going to put the modification onto the histone tail. All right, let's just give you in general what different types of modifications do. There are a whole list of different modifications on histone tails. And every one of them seem to have a specialized function for themselves. Okay, so when you look at the interface nucleus and look at the regions of the genome that are heterochromatic versus euchromatic, okay, where euchromatin is mostly inactive and condensed, and heterochromatin is mostly inactive and condensed, and euchromatin is active and open, and say what type of histone modifications I actually see at these regions, what you will find is that a methylation of lysine 9 on H3, which we would shorten as H3K9 methyl 3, okay? That is usually found in heterochromatin regions. Methylation of lysine 27 on H3, H3K27 methyl 3, is also found in heterochromatin. But we cannot generalize that all methylation is found in heterochromatin because you can see here that the methylation of histone H3 at lysine 4, H3K4 trimethylation, is actually found in euchromatic regions, right? And we can largely generalize the fact that the acetylation is mostly found in euchromatic regions, as well as serine 10 phosphorylation, though this modification is also specific to mitosis. So what this is telling you is that each specific modification can have a different function, right? And it is the reader of the function that is going to do the downstream process. So whatever the reader protein is for these modification is going to decide what the structure or the function of that modification is going to be. All right, let's talk about H3K9 trimethylation in a little bit more detail. This modification is found in heterochromatin. It actually mediates formation of heterochromatin. It mediates compaction and silencing of transcription. Okay? So it happens that certain regions of the genome needs to be silenced or compacted. What is going to happen is that you're going to have these nucleosomes in the genome, and an enzyme called H3K9 methyltransferase, so this is a methylase, right, is going to come in, and it's going to methylate the H3K9. So the methylation, it puts 3 methylation groups on the same lysine, methyl groups on the same lysine. Now, these methyl groups on lysine 9 serve as a binding site for a protein called heterochromatin protein 1, HP1. Okay, it's very imaginatively named. And this HP1 protein has a protein domain called chromodomain that binds to methylated H3K9. Its structure is such that it only binds to methylated H3K9. It doesn't bind to it when it's not methylated. Okay? So when HP1 binds to these methyl groups, it also has a different region of the protein domain that helps it to oligomerize. Okay? So it binds to each other. So you have a protein binding the methylated lysine tails, and it's going to bind to each other. What you're going to have is that everything is going to start clumping together. Okay? You're going to have oligomerization. So that means that it's going to start condensing. Does that make sense? And you can see here that in this case, the function of H3K9 methylation is to help the chromatin condense, right? All right, and here's a question for you then. If there is a mutation that abolishes the interaction between HP1 proteins, okay, what would be the effect on heterochromatin compaction? Go ahead. It would be more open, right? Because it's not going to be able to oligomerize, and you're going to have more open chromatin. So these are the types of questions that we ask in midterms, because it's going to test your understanding of the concept. So pay attention and try in your mind what kind of other questions we could ask about this process, right? And it will help you actually learn the concept behind it. Any questions about this? All right. Due to the mechanism by which the heterochromatin proteins interact with the modifications, the heterochromatin can actually spread from the initiating side to the other sides linearly. Okay? And how does that happen? So here's the mechanism by which the HP1 mediated heterochromatin can spread. So first, we actually have sites that are going to recruit the methyltransferase complex. Okay, so that's H3K9 histone methyltransferase, H3K9 HMT, to a site. So that HMT is going to put the methyl 3 on H3K9, and that is going to recruit the HP1 protein to bind here. The HP1 protein, in fact, itself actually can interact with the H3K9 HMT and recruit it to the region. Now, when HMT is recruited to this site, what it is going to do is it's going to look for these histone tails that are not methylated, and it's going to put a methyl 3 on the neighboring nucleosome. And now, when that happens, a new HP1 can come in and bind to it, right? So it's basically kind of like this. The first step is that you actually have a histone tail that's sticking out at a nucleosome. So you have H3K9 methyl 3. The HMT puts this methyl 3, right? And this methyl 3 is bound by HP1. And what it does is it interacts physically, right? Proteins can interact with each other physically through surfaces, right? And it's going to bring more HMT that interact. And what it is going to do is it's going to put more methyl 3, which is going to be bound by more HP1. That is going to bring more HMT, which is going to put more methyl 3. And that's going to bind more HP1. And it's going to spread continuously, okay? Now, it spreads until it encounters a certain boundary element where it cannot spread anymore. So these boundary elements is going to stop the spreading. So if the heterochromatin is spread until a boundary element, the other side of this element is going to contain chromatin that is going to be active, and the side that is containing the methyl 3 is going to be heterochromatinized, right? Any questions about heterochromatin spreading? All right. This is yeah, go ahead.
How much energy does this process require?
I have no idea. It will definitely require some energy. Some of these proteins are ATP dependent. Some aren't, and it depends on if the process is ATP dependent or not. The histone modifying complexes in general are not ATPase dependent. So the methylases are not ATP dependent. But there's another chromatin modifying complexes, which we term chromatin remodeling complexes, that literally go in and disrupt the histones, and that process is extremely energy dependent, and they are ATP dependent complexes. Among the histone modifiers, the ones that I'm aware of are not ATP dependent, so they probably do not spend a lot of energy, more than what would require to shuttle them in the nucleus, etc. But there might be cases that are ATP dependent. That's why the reason why I said I have no idea how much it would take. Any other question? All right. So that's how we spread. Where is heterochromatin? Heterochromatin are in the regions of the genome that need to be absolutely transcriptionally silent, and in specialized structures on chromosomes, which we actually mentioned at the beginning of the lecture. And these heterochromatin sites are pericentromeric regions. Pericentromeric means the regions that are right next to the centromeres, and telomeres. So the centromere is defined as the region of the chromosome that controls the movement of the chromosome during a cell division, because it helps form this protein structure called the kinetochore that attaches the centromere to mitotic spindles. Okay? And this structure needs to be pretty compact and strong. And that's why we see a lot of heterochromatin right around there. And the telomeres are the ends of chromosomes with repetitive sequence, and they are maintained by a protein complex called telomerase, which adds short oligos to the end and protects it from degrading every cell cycle. We're going to go into more detail in the later lectures. But what I'm trying to tell you here is that these are certain regions of the chromosomes that are heterochromatinized. And another region of the genome that is typically heterochromatinized and silenced are transposons and repetitive sequences. Why would it be important to silence transposons? Some of you guys were in the last lecture. Come on.
Like, if it's not where it should be, you could, like, transcribe some mutated.
A little bit more clear?
Like, let's say the transposon jumps to the middle of the gene, and you don't silence that. When you transcribe it as a protein, it's
Mm-hmm.
mutated.
That's about the lines what I'm thinking. So if it's disruptor, you want to keep it quiet, right? But let's imagine beforehand. What you want to do is you want to prevent it to be disruptive to begin with, right? What is the first step of transposition reactions? What's the first step of a transposon jumping? Come on, come on. You reviewed the lecture. I gave the whole lecture about learning. What's the first step of a transposon jumping? What does it need? What protein does it need? What protein is required for a transposon jumping? Remember, biologists name their proteins really boringly. Transposase. Where is transposase encoded from? Transposon itself, right? The open reading frame in the transposon. Well, if you can't shut off transcription of that transposase to begin with, then you're not going to have problems of the transposons to jumping around, right? So that's why a lot of the repetitive DNA and the transposons are actually silenced. It's in our own interest to keep them chromatinized, very compact, and silent, to heterochromatize them. All right. And other regions of the chromosome that I said is one of the regions of the chromosome that are heterochromatinized are telomeres and subtelomeric regions. Subtelomeric means regions that are close to the telomeres. In yeast and humans, this is done by a complex of proteins called the SIR proteins. Those are They are named after silent information regulator because they actually form heterochromatin in certain regions of the genome. Okay. So what we're looking here is we are looking at a my thing just crashed. All right, so what we're looking here is an immunofluorescence analysis of SIR proteins. Immunofluorescence analysis of SIR proteins requires this. Similar type of concepts. What you do is you are interested in where a protein is in the nucleus, okay? So the first thing that you do is you generate antibodies that are going to specifically bind to that protein. So we're interested in where the SIR protein is. And what you want to do is the first thing is that you generate antibodies that are specifically going to bind to that protein. So next, what you do is you use, and this is called a primary antibody, because it's going to primarily bind to the target, okay? And this is the target. You're going to use a secondary antibody that is going to recognize the heavy chain, the constant chain of the primary antibody. And what you're going to do again to this secondary antibody is that you're going to conjugate it to a fluorophore. And you can do these through chemical reactions, okay? And let's say that this fluorophore is a green or red or whatever color fluorophore that is available, okay? So next what you do is you take your nucleus, you basically fix the nucleus with some kind of chemical fixer that's going to stop all the dynamic processes that are happening, okay? So you're taking a snapshot of the nucleus. Next, you're going to take your primary antibody and you're going to permeolize your cell membranes, and you're going to put the primary antibody with the nucleus, and you're going to incubate that. It's kind of like hybridization, right? You're going to let the antibody find its target. It's going to go seek its target, it's going to find it, and it's going to bind to it because it has affinity to it. Next, you're going to take your secondary antibody, and you're going to send it afterwards, okay? And that's going to go find its target and bind. And when it binds to the primary antibody, and you take that whole thing under the microscope and look, wherever you see that fluorophore, wherever you see that green signal, is where your protein is. Does that make sense? It's going to come up over and over again. This is protein immunofluorescence, okay? Analysis of immunofluorescence. Now, in this experiment, what they've done is they've looked at where the SIR3 protein is. This is one of the proteins in the SIR complex that binds to the telomeres, okay? And at the same time, what they did is they did a DNA fish on the same sample, right? And in this DNA fish experiment, they used telomeric sequences to make probes that are going to bind to the telomeric sequences, right? And what that's going to give you is give you where the telomere is, right? Because this is DNA fish, where you use the telomeric DNA as probes. And what you can do is you can see, are these two signals overlapping? If they're overlapping, what is that going to tell you? How do you interpret that result? You've done a protein immunofluorescence to look at where SIR3 is. You're doing a DNA fish to look at where the telomere is. And you're overlaying two signals and looking if they're overlapping or not. If it's overlapping, what does that tell you? Speak up. They are co-localizing. That's what we call them, co-localizing. They're at the same site, right? It's telling you that the SIR3 is binding probably to the telomeres. Very good. All right. So how do these SIR proteins work? At the very core of it, there are three proteins that are called SIR2, SIR3, and SIR4. SIR2 is a histone deacetylase. What it does is it removes the acetyl groups from the N-terminal tails of the histones. Okay? And what that does is it provides a binding site for the SIR3 and SIR4 proteins, which like to bind to histone tails that do not have the modification, right? Remember, we said the histone modifications, one of the role could be kicking out certain proteins from the chromatin. In this case, the histone modification acetylation, the function of it is to kick out SIR3 and SIR4 from the chromatin. Okay? So now, when SIR2 removes the acetyl groups from the tails, the SIR3 and SIR4 say, oh, that's good, now I can go and bind to it, right? And they go and bind to it. They also have the same type of property as HP1, in which that they can interact with each other. So the more SIR3 and SIR4 you have, the more condensed and your chromatin is going to be. All right. Here is the question. Very similar to HP1, right? There seems to be a spreading mechanism here as well. That is, the SIR3 and SIR4 proteins actually interact with SIR2 and bring it to the region so the SIR2 can deacetylate more tails, and these guys are going to bind more, and it's going to bring more SIR2, and it's going to deacetylase more, right? There's like a positive feedback that's going to help for these guys to spread. SIR2 is the histone deacetylase, and SIR4 binds to H4 tails, unacetylated H4 tails. It's very similar to HP1, right? HP1 spreading. What would be the protein counterpart for SIR2 in that example? Speak up. HMT. Yes. In this case, you're removing the modification for the other protein to bind. In the HMT cases, you're putting the modification for the other protein to bind. Conceptually very similar, but details are not exactly the same. What would be the counterpart for SIR4? Speak up. HP1, because that is the reader. Right? That's the reader protein that's binding to the methyl 3 in the case of HP1. And the reader protein here is SER4. But what it reads is not the presence of the modification. It's actually reading the absence of the modification. Right? Any questions about SER proteins? All right. So the histone proteins in the genome can come in various flavors, okay? What we call the regular ones that you find all over the genome, what we call them are the canonical histones. And there are these histone proteins that are slightly different from the canonical histones. And these are called histone variants, okay? And these histone variants are histone proteins that go to specific regions of the genome to accomplish specific functions. So one example of a histone variant is called centromeric H3. So this is a histone H3 that looks very much like a histone H3, but has a few amino acids differences, and it's only deposited into the centromeres, and it functions at the centromeres. So the centromeres in humans look something like this. You guys remember microsatellites? You should. They are found at the pericentromeric regions that are close to the centromeres. The centromere is the region where the kinetochore is going to form and attach to the spindle, and it contains lots of nucleosomes, okay? And these nucleosomes, the light blue ones here I indicated will contain canonical histones. It's going to contain the regular H3. And it will also have histones that are scattered around the centromere, the nucleosomes scattered around the centromere that are going to contain this histone variant called CEN-H3, centromeric H3. And that is going to be the specialized histone that's going to form the nucleosomes. The CEN-H3, one of the functions that it has, it serves as a binding site for the kinetochore proteins. The kinetochore proteins bind to the centromeric H3, and they also have sites to bind to the mitotic spindles, okay? And that's how the centromere is going to connect to the mitotic spindles. Does that make sense? All right, so here is an example of a histone variant that goes to a special region of the genome and for a special function. Okay, here's a question. What if the centromeric H3 got deposited into another place on the chromosome that is not the centromere? What might happen? It might form a kinetochore. And let's say that it's catastrophic. It formed the kinetochore. It attached to the mitotic spindles. What would the fate of that chromosome be? Now that chromosome has two kinetochore attachment sites. That's not good for a monocentromeric organism, such as the humans, where we want one centromere per chromosome. That is not the rule across all eukaryotes. There are actually eukaryotes with chromosomes with multiple centromeres, but they have mechanisms to prevent any catastrophe. But in us, in humans, two centromeres is no good. I mean, two kinetochores is no good. Why? Think physical. The chromosome is basically kind of a rope. You have two of them stuck to each other at the centromeres with the kinetochores. They're going to attach to the spindles, and they're going to pull when the daughter cells are separating. What might happen? They can separate. Actually, the mitotic machinery is so strong, it is going to separate those. It's destined to separate. That's no good. What is it?
You have like an unequal distribution.
Due to?
It's just separated badly.
They are going to break. The reason why they might break is, imagine, let me see if I can draw. So in monocentric organisms, what you want to do is you want to have a single centromere that is attached to the spindle so these guys can go in their own ways, right? So if you have a second kinetochore, there is no mechanism to make sure that that kinetochore is attached to the same spindles, okay, in the same side. So if they are attached on the same side, there is going to be no problem because these chromosomes are going to be able to segregate. But imagine now that they are not, okay? So they attach to the wrong side. These spindles are really strong. What will happen is that this is going to pull it towards that side, but these spindles are going to pull the chromosome to this side. That one is going to be pulled on that side. This one is going to be pulled on that side. And the result of that is the chromosome will break somewhere around here, right? And what you're going to have is a chromosome breakage with ends that are not telomeres. So when you have ends that are hanging around, the cell is going to say something wrong is going on, right? And if that happens, it's going to use repair machineries to repair, and we're going to learn about that in the next lecture. All right. So the interface chromatin structure also contains loops of chromatin fibers. Okay, so we know that they exist. We don't know how many there are. They are kind of dynamic, but when you look on the microscope or do molecular biology experiments, you see these loops. Okay, one evidence for these loops to exist comes from DNA fish experiments. So what people have done is they made probes to different regions of the chromosome. Let's say the region A and B and C and D, E, F, G, H. Okay? And they hybridized the chromosomes with these probes. What the researcher knows is that in the linear space, because chromosomes are linear structures, you can actually give a coordinate, right, from 1, 2, 3, 4, 5, etc., etc. And that would be linear coordinates. In the linear space, two probes can be far from each other, but when you look at in the three-dimensional space, they are close to each other. And the way that they can come close to is through looping, right? So when the researchers looked at these two probes, that are far away in the linear space but very close to each other, they said, okay, these are they are at the base of the loops, very close to each other, right? And then these two probes are more close to each other, even though in the linear space, that probe C is closer to D. Okay? So what they can infer are the presence of these loops because of these experiments. Does that make sense? So even if you can't see the loop, like nicely cartoon drawn like our pictures, you can infer that structure through DNA fish experiments. Any questions? All right. What is the molecular proteins that hold these loops together, that hold the loops structure? These are called it's a family of protein complexes. They are called the structural maintenance of chromosomes family of proteins. One example of this is cohesin, which keeps the sister chromatids together during the S phase. The other one is condensin. It got its nice imaginative name through its function to condense chromosomes during mitosis through loops. The chromosomes are long threads, and the way that you can condense them and shrink them is you can generate loops, right? And condensin helps generate those loops. And the cohesin helps keep sister chromatids together. They look something like this. There are two proteins called SMC2 and SMC4. They heterodimerize, and a third protein called Cleason bridge protein closes this loop, and you basically have a ring-like structure. And that ring is going to hold the two chromosomal sites together, forming the loops. Now, the loops can occur during mitosis, and it occurs during interphase. In interphase, it can occur intramolecularly. Intramolecular means on the same molecule. What I'm meaning is on the same molecule of DNA. So the same chromosome can fold onto each other, forming a loop. Here, what you are having is that cohesin is acting intermolecularly, right? it is connecting two different chromosomes, in this case two sister chromatids, and here connecting two distant sites together. And we're going to talk much about these kind of loops and the function of them in gene regulation gene regulation lectures, where the two distant sites on the same chromosomes has to come together for a gene to be expressed. All right. So all of these different types of packing mechanisms and organization mechanisms, at the end, organize so that each chromosome lives on its own territory. So if you think of chromosomes like a bowl of soup, right, and it's basically spaghetti. Spaghetti is each chromosome. They're actually not intertwined with each other. Is that they're kind of segregated. Wouldn't it be amazing if you bought a bowl of soup where each of these noodles were separated? I'll totally freak out. But that's basically how the interface chromosomes look like. They segregate in their own territories. So here's an experiment where people have done is they made a fish probe to each individual chromosome, and they hybridize it to the interface nucleus. I believe these are chicken cells. Chicken fibroblast nucleus. Fibroblasts are kind of squished because they're muscles, right? They're long and elongated, so that nucleus doesn't look nice and round. So they're squished here, but what you can see is that each chromosome that is stained is kind of occupying its own territory with some mixing going on as well. All right. Why is this important, right? This could be a cause or a consequence of something that is going on in the nucleus, and this is one of the open research questions that a lot of people work on, including me as well. So I work on how chromosome structure regulates gene expression. But there are some clues where the chromosome structure and how this organization relates to gene expression. One of them is that when you look at where heterochromatin is in the nucleus, what you find is that it's mostly at the periphery of the nucleus. Okay? It's close to the membrane. How does it actually get there? So these nuclear membranes have proteins under the membrane that are called lamins. So these lamins are proteins that cover the membrane. It's kind of like form that kind of skeletal structure. And these lamin proteins also like to bind to heterochromatin. Okay? So the heterochromatin sites in the genome, not always, some certain regions that are called the lamin-associated domains, go towards the lamina, nuclear lamina. When they are there, they're silenced. If you actually prevent their sitting at the lamina, they start to get de-repressed. That means the genes under these domains are going to get starting to express. So their positioning into the lamina is important for the gene regulation to occur correctly. So here's just a little cartoon. Don't worry about the details here. Just see that there are lamin proteins under the nuclear membrane. And there are proteins that actually interact with the lamin proteins as well as proteins that are associated with the membrane that bring the heterochromatin, certain types of heterochromatin, to the nuclear lamina. So is this important? We think that it is important because if you mess up with the mechanisms that actually bring the heterochromatin to the periphery, is going to mess up with the gene regulation. One of the most drastic examples of this is the analysis of a mutation in the lamin protein. Okay? So what we're looking here is another immunofluorescence experiment. And this experiment, instead of using antibodies that are going to specifically bind to a protein, you're using antibodies that are going to specifically bind to a histone modification, right? And what this is going to allow you is, at the microscopic scale, to find out where these histone modifications are. So if you use an antibody that is conjugated to green and recognizes H3K9 methylation, but in normal cells, what you see is these green regions, and you know that those regions are the heterochromatic regions of the genome because it has H3K9 methyl 3. And if you look at a mutant where the lamin A is mutated through a silent point mutation, that produces a incorrect mRNA splicing site and causing a deletion of 50 amino acids from itself, lamin A. What you can see is in those cells, first of all, the nuclear structure is messed up, right? It doesn't look nice and round anymore, as well as the heterochromatin is messed up. That you see actually less H3K9 methylation, and you do not see these domains, heterochromatin domains, that are actually juxtaposed to the nuclear membrane. And you can see that other type of histone modifications are also affected, but not the same way as H3K9 methylation. What that's telling you is that lamin A mutation and the problems in the nuclear envelope is causing some problems the way that the heterochromatin is organized and the way that the heterochromatin marks are made. So what happens? This mutation is actually isolated from children that show early aging syndrome. So this is Hutchinson-Gilford progeria syndrome. So these children progress through their lives very quickly and typically die like their mid-teens, early 20s due to diseases that usually kill older people. Okay, so this type of that mutation gives such a drastic phenotype. So there is now an initiative at NIH to go after these mutations and find out how you can actually correct the phenotypes. All right, with that, I'm going to stop. Again, there are some useful videos to watch and study your study questions, and I'll see you on Wednesday.